These talks are sponsored by the California Institute of Regenerative Medicine, which funds excelling students in our biotechnology program in 10-month internships around the state of California, where they further the frontiers of stem cell biology and regenerative medicine. Dr. David Schaffer has had a rapid and impressive career, and his many accolades and awards precede him. They include being named a Top 100 Innovator by the Technology Review. He is a professor of chemical and biological engineering, bioengineering, and neuroscience at the University of California, Berkeley. He also acts as the director of the Berkeley Stem Cell Center, which brings together researchers from the Lawrence Berkeley National Labs, University of Berkeley, and Corey, the Oakland Research Institute. He graduated from Stanford University, then earned his Ph.D. at MIT in chemical engineering. He then joined Fred Gage at the Salk Institute before moving to Berkeley in 1999, where he applies engineering principles to stem cell biology to further our ability to work with neurons. So without further ado, David Schaffer.
I know that we have a diverse background of people who know a lot about biology and people who maybe don't have as strong a background, maybe come from other walks of life. And so please bear with me for a minute or two while I provide you a bit of an introduction into what a cell is initially, so that we're all on the same page. So when I start talking about stem cells, everybody can start raising their hands and asking questions about those cells in particular. So a cell we can define as a fundamental unit of life. And the majority of cells on Earth are simply a single cell, such as a bacterium or a yeast cell, a fungus, for example. And these cells are made up of a number of building blocks, DNA, proteins, RNA, fats or lipids, metabolites. And every once in a while, about 10 to the 13th or 10 to the 14th of them will get together and make up a human being. So this is the number of cells inside of every one of us. So in addition, these cells serve a number of different functions, and not every single cell in our body is obviously identical. So depending upon how finely you slice them, we have something on the order of around 200 different types of mature cells inside the body. And these can range from skin cells, cells that line the stomach, liver cells, or neurons, for example, shown over here. And furthermore, there are a number of ways that cells can begin to misfunction in disease or injury. And when a particular type of cell all of a sudden begins to die, these types of diseases occur. And these are serious healthcare problems within the United States. Just to provide you with a few numbers, there are around 4 million patients with Alzheimer's disease right now, 1.5 million with Parkinson's disease, 3 to 4 million with stroke. And I'd like to actually ask, everybody who knows somebody who has Parkinson's disease, can you raise your hand? Keep your hand up. Alzheimer's disease and stroke. So as you can see, just even looking around the room, these are major issues that face our country right now, and there are currently no cures for any of these diseases. They're simply palliative treatments or treatments that really address the symptom rather than the underlying disease. And furthermore, in each one of these types of diseases, large populations of cells are actually lost. So for example, if you lose a population of cells in your brain called a cortical neuron, you end up developing symptoms of Alzheimer's disease. If you lose a population of cells called dopaminergic neurons within a region of the brain, you end up suffering from the symptoms of Parkinson's disease. And by the time somebody goes to the clinic and begins to present with Parkinsonian symptoms, they've typically lost 80% of these cells already. So in many cases, it's not enough to simply halt the progression of the disease. You actually need to develop ways to replace the cells that have been lost. So that's where a stem cell comes in. So if you start out with a fertilized egg, a zygote, this divides in 2 to the nth fashion, 2, 4, 8, 16, 32, until you end up with a small ball of cells, a very early stage embryo that has around 64 cells. And this early stage embryo has 2 major cell types, extraembryonic cells that will form the placenta, for example, and the so-called inner cell mass. So the in vitro fertilization process began about 33 years ago or so. The first IVF baby is around 33 years old. And when somebody conducts IVF, they typically harvest around 8 embryos or so, or generate around 8 embryos. And they typically freeze down maybe 6 or 5 of them, and then implant around 2 to 3. And that's one of the reasons why often in vitro fertilization results in twins or triplets. But what that means is that there are around 5 embryos that are frozen down for every time there's an attempt at doing an IVF. And if you add that up over around 30 years or so, there are around 400,000 frozen embryos across the country right now. A lot of the parents who tried to have kids 33 years ago are now well into their 60s. They're done with the embryos. They don't actually need them. So some of them have actually donated these embryos to science. And if you actually isolate this inner cell mass, you can generate an embryonic stem cell. And this cell is known as a pluripotent cell, meaning it has the ability to generate every single cell type within the adult body. Furthermore, this cell can specialize and, for example, turn into a tissue-specific stem cell, such as a skin stem cell or a blood stem cell or a neural stem cell. And these are multipotent cells that have the ability to turn into a few cell types, but not all the cell types in the body. And so, for example, neural stem cells that we work with have the ability to differentiate into cell types in your brains known as glial cells, astrocytes and oligodendrocytes, as well as kind of the business or functional end of the nervous system, neurons. So furthermore, stem cells don't just occur during the development of adult organisms. You actually have pockets or niches of stem cells distributed throughout your bodies. And so, for example, if you actually analyze tissue within the adult brain, it actually harbors neural stem cells, which, like a neural stem cell that arises during the development of adults, can continue to give rise to neurons and astrocytes and oligodendrocytes throughout human life. So I'll be talking about those cells for the rest of today. But regardless of where the stem cell comes from, it really shares two defining properties. One is the ability to kind of zip around this circle to actually make more of itself, to divide in an immature state, a process known as self-renewal. And the second to move from the left hand to the right-hand side of the slide to differentiate or acquire specific function, for example, the cells that are necessary for the function of the adult nervous system. So the idea then is that these cells can be potentially used to create new cells to treat disease. So the way it would work would be that you would isolate or derive a stem cell, whether it's an embryonic stem cell or an adult neural stem cell. You can then expand it, induce it to self-renew to create many more copies of itself. You can then differentiate these into tissues or tissue equivalents, differentiated functional cell types, and these can then be implanted into a site of disease or injury in order to try to repopulate tissues from the devastating effects of those diseases and injuries, and thereby repair the function of these adult tissues. So what I'm going to be talking about for the rest of the day is starting out with some definitions and furthermore moving on to some basic biology. And towards the end, I'm going to be talking about some of our results. The majority of what I'll be talking about today is more an historical overview of the field rather than the specific work that we're doing within our lab. But I will be talking towards the end about some of our efforts to harness these cells for tissue repair. So if you take a look at the development of the nervous system, and for the remainder of the talk, I'm really going to be focusing on the brain in particular. So within human beings, the largest number of neurons are actually born between around 5 weeks to 5 months after conception. And during this time, there's a peak rate of around 250,000 new neurons created per minute within the developing brain. And so this is obviously responsible for the massive growth of the brain from the time of conception all the way to the time of birth, roughly. And furthermore, the cells that are responsible for this are the neural stem cells. So for example, these cells, as they're dividing from, or as they're differentiating or specializing from the embryonic stem cell, they create these neural stem cells that very, very rapidly divide and are responsible for the growth of the brain. So furthermore, these cells actually, while they're very prevalent in early developmental stages, become more and more rare as time goes on. So for example, if you take an embryonic day 8 rat, I'm going to be hopping around a couple of different organisms or model systems, but 8 days after a rat is fertilized, or a rat egg is fertilized, around 50% of the cells that you isolate from the nervous system of that rat are actually stem cells. They have the ability to divide and to generate multiple cell types, such as neurons and astrocytes. If you wait to around day 10, within a mouse at least, only about 20 or 5 to 20% of the cells are neural stem cells. And then as time goes on, if you wait to embryonic day 12, only 10%. Postnatal day 1, the day after birth, only around 1% of the cells are actually neural stem cells. And the idea is that as the brain and the spinal cord become more and more mature, there's less and less need to create new neurons, to create new cells. So this idea actually led to a dogma that has really dominated the field over the past 100 years. And this is really the father of neuroscience, a Spanish person named Ramon y Cajal, who won the Nobel Prize in 1916 or so for actually doing this, generating a number of images, staining neurons, and actually tracing the wiring of electrical connections inside of the brain. So within adult organisms, he never saw a situation in which there was a mature neuron that was actually in the process of dividing, the process of turning into two brand new cells. And based upon this, he came up with this idea that once development has ended, the fonts of growth and regeneration of axons and dendrites, the wiring of the nervous system, dry up irrevocably. In adult centers, nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated, and it's for the science of the future, if possible, to change this harsh decree. So how many people learned that once you're an adult, you can't actually add new neurons to the brain? So I learned that certainly in high school and college. And it turns out it's wrong. So I'm going to be providing you with a couple of pieces of data that led up to the field actually concluding that the adult brain can add new neurons. So the first piece of evidence came in the 1960s. This fellow named Joseph Altman, who was a professor at MIT, he actually injected some labeled nucleotides, where nucleotides are the molecules that get incorporated into DNA. These are sort of the monomers that make up the polymer of DNA. And by doing this, he was actually able to image cells that were undergoing cell division. And they took a look at these same cells that are shown here under the light microscope and said, hey, that looks like a neuron. This is evidence with an adult organism that you have cell division followed by that cell turning into a neuron. And based upon all of this evidence, he was actually largely ignored within the field. So if you fast forward a few more years, microscopy has gotten better. So we're no longer looking through a light microscope at cells. Now this is an electron microscope. And the same idea, they labeled a dividing cell, shown right here. There's the label telling us that that cell divided. And then used electron microscopy, and they said, hey, that really looks like a neuron now. It has a process, an axon, and it looks like that cell is communicating with other cells in the nervous system. So this evidence was actually largely ignored as well. So it took a quote-unquote lure organism to begin to kind of break this dogma. And within the 1980s, there was research conducted with songbirds, canaries roughly. And it was noticed that these animals actually do have very, very large numbers of neurons that are generated kind of periodically. It follows the calendar. So this is June, July, August, September, etc. And this, on the y-axis, is the number of newborn neurons within a couple of regions of the brain, shown up here. And you can see it peaks right around October and right around March. And this follows the mating season of these birds. So if you then analyze behaviorally how many new syllables these animals learn, how much more song they can acquire, it actually follows the birth of the new neurons. So the idea is that the males need to learn new song in order to attract a mate. That's such an evolutionarily important task that they're actually going to acquire new neurons to be able to do that. And once they're done, once they've acquired a mate, the cells actually die off during the intervening time. So there's this rapid turnover of neurons within the brain. So furthermore, this fellow actually proved that those neurons were actually integrating into the electrical circuitry of the brain and contributing towards the function of the brain. He actually stuck electrodes into the brain and took measurements upon those newborn cells and proved that a newborn cell, labeled right here, was actually electrophysiologically active. It was actually carrying current or conducting current. It was responsible for function within the brain. Okay, so this began to chip away at some of the dogma, and it took until the early 90s, actually, where several investigators really began to acquire definitive evidence proving that the adult brain can generate new neurons. So this happened in 1992 in one region of the brain. It actually happened in 1993 by my postdoctoral advisor. And they discovered or actually harvested cells from the brain and showed those cells could divide. And so they took up this marker, this labeled nucleotide, this little piece of DNA that was incorporated once a cell divided and had to copy its genome, had to double the amount of DNA present. And furthermore, that newly divided cell later turned out to turn into a neuron, and as a result, it acquired some markers, some stains that told us that the cell had differentiated into a neuron. So if you fast forward a few years, in the mid-1990s, it was then definitively established and proven that Ramon y Cajal, that father of neuroscience, was right most of the time. The majority of the brain lacks the ability to add new neurons. But he was wrong in a couple of places, a couple of rare places. So there's this one region called the subventricular zone, right around here in your brains, towards the forebrain. And this region contains a population of stem cells that would continuously divide. And the progeny, the daughters of those division events, will migrate a couple of centimeters in our brain, a few millimeters in a mouse brain, and will populate this other structure known as the olfactory bulb, so olfaction or smell. So the acquisition of these new cells is important for the ability of these animals to learn new smells. And if you actually delete these cells within a mouse, they lose the ability to recognize their pups, their children. So the sense of smell is incredibly important for a rodent. But there's this other region of the brain that's very, very important for us called the hippocampus. And if you zoom in on this hippocampus region, there's a little V-shaped structure right there. And this is a blow-up of it within an image that we acquired. And it has a few different populations of cells. Shown in green is a stain for a mature neuronal population. Shown in red is a very important population of cells called astrocytes. They provide structural support and a lot of nutritional support for the neurons. And furthermore, shown in blue here is a dye telling us that these cells are the stem cells. So these cells, which are relatively rare, will actually divide very rapidly, though, and will generate something on the order of 10,000 brand new neurons within your brains every day. So the way that this works is that these stem cells kind of reside on this edge between the neurons and the astrocytes, a region called the subgranular zone. And they divide, and then will migrate a couple of cell diameters, and then will extend their circuitry and integrate into the circuitry of the brain. You can isolate these cells as well to be able to study them. So shown here is an image of a population of immature cells. And the way that we analyze whether the cells are immature or beginning to differentiate, and if they're differentiating, what they're actually turning into, is to use stains, antibodies that are fluorescently labeled so that we can actually image them under a fluorescence microscope. So these cells stain positive, for example, for this marker that is called nestin. And this protein simply tells us that these cells are still in a very immature stem cell-like state. If you begin to add components to the cells, you can then induce their differentiation into the variety of cell types that appear within the adult brain. So for example, you get a mixture of oligodendrocytes, shown here in green, where oligodendrocytes are the cells that wrap themselves around neuronal processes and insulate the neurons so that they can better conduct electricity, kind of like plastic being wrapped around this wire. And patients who lose oligodendrocytes develop multiple sclerosis. They lose the ability to conduct electricity in the brain. And furthermore, there's another population of cells, shown here in red, these astrocytes, which I showed you right here a second ago. And finally, we have the business end of the nervous system, the actual neurons themselves that can be generated from these cells in culture. So now we have a situation in which it's been proven that rats and mice... Yes, question?
Thanks. How do the dyes know where to stick to these cells?
Your immune system creates very, very large collections or libraries of molecules called antibodies, which you're probably familiar with. So these antibodies are responsible for our ability to recognize very specifically, invading bacteria or viruses. It's part of our immune system, our ability to repel invading pathogens or organisms. So that's why you get vaccines. When you go to get a flu vaccine, you get an injection in your arm, you get exposed to a little kind of a weakened version of the flu that's circulating this year, and you raise antibodies against that flu antigen that you get injected with. So if you end up getting exposed to the actual flu, you have antibodies that can protect you from it. And these antibodies can bind to molecules such as proteins with a high degree of specificity. So what we have here is an antibody directed against a protein called neurofilament 200, which is a marker telling us that this cell is a neuron. So you can take that antibody, you can isolate it. There are ways to produce these in large quantities now. You add on a green tag, a fluorescent tag on it, and then it will actually label or stain the cell, and that'll tell you that this is definitively a neuron. And incidentally, the monoclonal antibody market, Genentech, that's what makes Genentech a company. They produce large amounts of monoclonal antibodies as drugs to recognize specific things for medical purposes. So the antibody market is around a $65 billion a year market in the healthcare field. But we simply use them at much lower scale within the lab, obviously. So we know now that at least rats and mice have the ability to create new neurons. And this doesn't say anything about human beings, right? So it was shown in 1998, again by my postdoctoral advisor, that the adult human brain does definitively add new neurons throughout life. So these were patients who were actually suffering from tumors in other regions of their body within Scandinavia. And within Scandinavia, a common medical procedure, a diagnostic procedure, is to inject one of those labeled nucleotides. And so it'll actually get localized and get incorporated into the DNA of all dividing cells. So the reason that they do that is that they can then biopsy the tumor, take a look at it under a microscope, and learn how rapidly the tumor is dividing. But as a byproduct for that, it actually labeled all other dividing cells within the body, including the newly dividing stem cells within the brain. So after these patients unfortunately passed away due to their cancers, they obtained tissue from these brains, and they could stain it and definitively show that a cell that divided while that patient was an adult, while they were suffering from their tumor, later actually turned into a neuron, and as a result acquired a marker like this one that told us that this cell was actually a neuron. So this definitively proves that you have the ability to create new neurons within your brains. And furthermore, that this is a highly conserved process that is undergone by all mammals that have actually been tested to date, everything from rodents all the way through primates. Okay, so another question actually is what cell type is it that creates these? So it's possible that there's a single neural stem cell that creates both neurons and astrocytes and oligodendrocytes, or another possibility is that there's actually a more mature cell, something that is perhaps already committed to turning into a neuron or already committed to turning into an astrocyte. And to be able to test where or how high on this totem pole these cells actually reside within the brain, investigators conducted a tracing experiment where all of these clusters of cells actually came from a single cell. And this is very recent work. And they found that a single cell could give rise to two stem cells. It could give rise to a stem cell plus a neuron plus an astrocyte, to a combination of a stem cell plus a neuron or a stem cell plus an astrocyte. So this was the first work that really definitively proved that there is an actual stem cell that exists within the brain. And it's not simply a collection of dividing neural precursors or astrocyte precursors. Furthermore, it was shown about 10 years ago that these cells actually do integrate into the circuitry of your brain. So after dividing, these cells extend their wiring, quite beautiful connections, and investigators began to hook up electrodes to these cells, began to stimulate them to see whether or not they could actually conduct current, could actually fire off an action potential, which is what a neuron does to communicate with the next neuron down the line, and they definitively proved that these newborn cells do so. So furthermore, another question is, how long do these cells stick around? I told you get around 10,000 new ones every single day. You obviously can't do that for the rest of your life because your brain doesn't have enough space, your cranium doesn't. So they labeled these cells, again, with that labeled little nucleotide, and then took animals, this is in mice, and analyzed what fraction of the cells that were present on day one actually stuck around for a long period of time. And this is about 2/3 of the lifespan of a mouse, so that's a very, very long time. So it turns out that the cells, a lot of them actually turn over or die, but around 20% of them actually persist for the life of the animal. And therefore, they're in a very good position to change the way that the brain functions all the way through adulthood. Okay, so if you take a look at these cells now, it turns out that they kind of live in sort of a hierarchy. So this is kind of the grandfather, the top of the totem pole as far as the stem cells go. This cell can differentiate and give rise to something that's kind of a precursor that has the ability to divide a lot. This in turn will differentiate a couple more steps and actually turn into that neuron. And there are a few markers that tell us which stage along this maturation process these cells exist. Okay, so this process of neurogenesis, or the generation of new neurons within adult organisms, is not fixed. It's not actually exactly 10,000 new cells in your brains every single day of your life. It's actually regulated by your decisions, by your behaviors, by your choices in life. So, for example, I'm going to go through some good news and some not as good news. So, it's been known for a long time in cognitive neuroscience that environmental enrichment, exposure to new signals, new cues, new experiences, actually enhances performance on learning and memory tests. So if you expose yourself to a rich environment versus a boring environment, you can actually learn better. You perform better in a number of behavioral tests. So the same kind of analogy exists with mice. So mice typically live in kind of a boring plastic cage with a lot of wood shavings in the bottom and not very much to do during the day. So investigators began to, again, this is my postdoctoral lab, ask the question whether enriching the environment of a rodent could actually affect neurogenesis. So they provided some pipes for the animals to run through, a ball for them to nudge around, a running wheel, which will become important in a minute, some better food sources, cookies and apples instead of rat chow, and then took a look at neurogenesis, the ability of these animals to create new neurons. So these are stains of the brain, and every single one of these dots corresponds to a new cell. So they analyzed how many cells had just divided one day after they did this labeling, this injection of the labeled DNA. And they had equal numbers of cells that had divided in a regular boring cage versus an enriched environment. But if you waited 4 weeks, twice as many cells actually survived for that long period of time. So that tells us our experiences actually change the rate at which new neurons are added to the brain. So they then took this kind of enriched environment and broke it down into a few components. So there's a control. These are the boring cages. There's an animal that was exposed to a situation where they were learning, an animal that was exposed to a situation where they had enforced exercise. They had no choice but to exercise. And then kind of going back to this running wheel, the animals were given the opportunity to voluntarily exercise. And then if you take a look at the number of newborn cells on day 1 and then what fraction of them or how many of them stuck around for 4 weeks, you'll notice that there's a significant increase in the number of newborn cells for the animals that were in this voluntary exercise situation, the running wheel situation. And then if you follow that out, a large number of cells actually persisted. So when Henriette van Praag, the investigator who discovered this, came up with these results, she quite literally went out and bought a pair of Nikes and started running on the beach in San Diego. So another interesting thing, actually, well, I'll come back to it in a second, actually. So another question is, so this behavioral choice, this voluntary running actually creates more neurons. Does this have an impact on the function of the brain? And so they analyzed learning. So you've all heard about the mouse in the maze cliche. So this is a measure of how long an animal takes to get through a maze, something called a Morse water maze. And furthermore, how long of a path they actually traverse before they get to the solution. So animals within the boring environment, and you'll see that as time goes on, as the number of days of testing of these animals goes on, they learn. It takes less time for them to make it through the maze. But the animals in the boring environment take much longer in every single situation. So exercise enhances neurogenesis, enhances the generation of new neurons, and it enhances learning within these animals. Okay, so that is one piece of good news. Exercise and environmental enrichment, enhanced neurogenesis, these are choices that we can make on a daily basis. And there are a couple of things that are actually not as good news. So, for example, age is a major factor that actually leads to the decline or the decrease in the generation of new neurons. So this is a rat. 27 months is a pretty old rat, but even between 6 and 12 months, this is kind of a middle-aged animal, there's a significant drop in the birth of new neurons. And furthermore, stress is a very potent inhibitor of neurogenesis. There have been numbers of ways in which animals have been stressed out, all the way from restraint stress, being held in one spot, to being exposed to the scent of a predator, all the way through the actual injection of glucocorticoids, the hormones that mediate stress effects. And in each one of these cases, there's actually a decrease in neurogenesis. So in retrospect, investigators actually used this to kind of explain this result, that these were the animals that were forced to exercise. They had no benefit. So the idea is if you're forcing somebody to exercise, you're stressing them out, and that actually counteracts the effects of voluntary exercise or some of the beneficial effects of exercise. So if you're going to exercise, you kind of got to want to do it. Yes, question.
So the exercise, you're saying that it's good for stem cell development or growing new cells.
Yes.
Is it just the activity or more oxygen in your brain because you're heavily breathing?
That's a great question. I'm actually going to be talking about molecular signals in a few minutes. So we really are just beginning to learn about the molecules that mediate these effects. So, for example, we've discovered recently a couple of molecules that increase with age in concentration in the brain, and that appears to actually keep the stem cells quiescent or repress their division. And there are other molecules that perhaps mediate some of the effects of exercise and environmental enrichment on stem cell function, but we don't know the answer to that just yet. Okay, so now the field has a very large number of correlations where, for example, stress has been associated in the cognitive neuroscience field with reduced or impaired learning, and it also reduces neurogenesis. Aging is a factor that leads to reduced learning in some cases, and it also represses neurogenesis. Radiation. So there have been a number of situations or a number of studies in which kids who are suffering from brain tumors, from gliomas, get exposed to radiation. And it's been shown in these cases that they never recover. They actually suffer lifelong deficits in learning and memory. And an investigator, a colleague of mine at Stanford, discovered that radiation is actually one of the most potent repressors of neural stem cells in the brain. Completely shuts them off permanently. Yes?
So if radiation shuts them off, what about radiologists because they're near radiation all day?
It depends upon dosage. So you need a certain amount of concentrated dosage of radiation going into the brain in order to begin to suffer the effects of radiation. So as long as you control the dosage, and we get exposed to radiation all the time, all you have to do is go outside and get exposed to the sun, or take an airplane flight, because then you're going way up into the atmosphere, and some of the shielding that the atmosphere usually, or protective effects that the atmosphere exerts on radiation, get reduced. So you get exposed to some radiation in an airplane. And obviously X-rays. So all of these are pretty low levels of radiation compared to this. So environmental enrichment has been correlated with enhanced learning and enhanced mood, and that results in enhanced neurogenesis as well. If somebody goes to a psychologist and complains of depression, one of the recommendations is to begin exercise, because this has been shown to definitively enhance mood, and it also is correlated with enhanced neurogenesis. SSRIs, anybody familiar? Yeah, exactly. Selective serotonin reuptake inhibitors, otherwise known as Prozac. So obviously the goal here is to enhance mood, and this has been associated with enhanced learning as well. There was actually a study published last year which indicated that sex enhances neurogenesis. I won't comment about what it does to mood. There also have been a number of studies that have taken different strains of laboratory mice which have different kind of genomes, different genome sequences, and have showed that each one of these genetic backgrounds results in a different level of, at steady state, birth of new neurons, and that level of new neuron growth actually correlates with the performance of these animals on learning and memory tests. So there's also a genetic component to this effect. Okay, so I've talked a lot about behavioral changes for the stem cells, the birth of new cells, what affects their survival and their maintenance. I haven't actually talked very much about what they do. So to be able to actually definitively study the role that these cells play within adult organisms, the most straightforward thing to do is to kind of knock them out, to ablate them. And very recently, technologies have been developed to be able to do this very specifically within a mouse. So this is some work, again, out of my postdoctoral lab. And I'm not going to go into the details of this study, but what they did was they actually showed that if you kill these cells, if you ablate them, these rare stem cells within the brain, the animals suffer deficits in adult learning and memory tasks. So it does appear that these cells are necessary for some learning and memory capabilities within the adult brain that are hippocampally dependent. Okay, so the next question that I want to focus on for a few minutes is, so we have behavior out here. We have the effects of behavior on stem cell function, and what are the molecules that actually mediate this? What are the signals that the body utilizes to control the function of these cells? So if you zoom in now, and this is a cartoon that was generated in the lab of somebody at UCSF, Arturo Alvarez-Buya, and this is kind of a cartoon mock-up of where the stem cells reside within the brain, or at least one of the two populations, that SVZ population. So these cells reside within a very geometrically complex region. Shown in blue, they come into contact with each other. They come into contact with a couple of differentiated populations of cells, so-called appendable cells, astrocytes, neuroblasts. They touch the cerebral spinal fluid, kind of the plumbing of the adult nervous system, the fluid that circulates around the brain and spinal cord. And they also come into contact with blood vessels. So each one of these interfaces represents a place where molecules can be transmitted from the environment to the stem cell in order to tell them what to do. And so these cells, in general, stem cells within the body, reside within anatomically specialized regions called niches, or microenvironments. And these microenvironments provide tightly regulated molecular signals that precisely control behaviors of stem cells, because you have to very precisely control that behavior throughout life to enable that stem cell to repopulate your brain with new neurons, or repopulate your blood system using blood stem cells, or enable you to lose skin every day and all of a sudden have a new layer of skin ready to take its place. So all these stem cells are very precisely controlled by the body in order to carry out these various functions. And these are the molecules that do it. So they're small molecules, things like retinoic acid or even ions. So electrostatics or electrical signals can regulate the function of the stem cell. Some small proteins known as growth factors or morphogens. Most cells in our body are actually enmeshed in a cage of proteins called the extracellular matrix. And furthermore, they're specialized cells that actually come into contact with the stem cell in the niche, and information can kind of be transmitted across that interface between the two cells. So I want to give you a couple of examples from our own work. And so around 12 years ago or so, we discovered that this particular molecule is a regulator of neural stem cell division within the brain. Does anybody play video games? So scientists sometimes have a bad sense of humor, and so the person who discovered this molecule actually named it after the video game. So it's a small protein that was discovered within the developing nervous system, and its role within the adult nervous system hadn't really been studied. So we actually began to investigate this protein, and last decade, around 10 years ago or so, we discovered that it actually serves as a mitogen, where mitogen is simply a word that means that it causes the neural stem cells to divide. So if you add this molecule to the stem cells in culture within a dish, you end up increasing the number of cells in a dose and time-dependent fashion. And then if you actually deliver the gene encoding this molecule into the brain so that you end up producing more of it within the hippocampus, you actually increase the overall number of cells in the brain, you roughly double it, and then if you wait a few weeks, you actually increase the number of newborn cells that actually turn into neurons. So by infusing this molecule into the brain, you can actually upregulate neurogenesis. So furthermore, I'm going to skip past that, it was not very widely known exactly what molecules take those stem cells and regulate their differentiation into a new neuron. So the signals that were responsible for the differentiation of those cells weren't very well characterized. So we began to study, this is just about 5 years ago or so, these pairs of molecules called Fs and Ephrins. So this is a molecular signal that's present on one cell, and it binds to a receptor, kind of an antenna, on the surface of another cell, and transmits information at the junction between the two cells. So we found that this one particular ligand, this signal, was present on the surface of astrocytes within the hippocampus, this important supporting cell type. And furthermore, the receptor, the antenna for that molecular signal, was present on the surface of the neural stem cells. And furthermore, these astrocytes came into contact with the neural stem cells. So the astrocytes are actually part of the niche or part of the microenvironment. So if we ended up infusing this molecule into the brain, and then you actually count the number of newborn cells that turn into neurons, where 100% means that all of the newborn cells turn into neurons, 0% means none of them. If we actually infuse just some saline just to control a solution, we end up having around 40% of the cells, the newborn cells, turning into neurons over a 5-day period. But if we infuse this molecule, that number roughly doubles to around 80%. So this tells us that we've discovered a molecule that stimulates the generation of new neurons from those neural stem cells. So it's not enough, if you're trying to establish the biological function of a molecule, it's not enough to simply add it and show you have an effect. You have to show that if you erase it, you lose that effect. So we then used a technique called RNA interference to actually knock down the production of this signal from the astrocytes within the brain. So this is a stain of the hippocampus showing that we have lots of the Ephrin, this signal within the astrocytes. And if we add this shRNA to induce this mechanism called RNA interference, we actually wipe out the expression of Ephrin within the brain. And does that have consequences? So now this is the fraction of newborn cells that have turned into neurons within this experiment. And then by knocking out the Ephrin, we actually knock that down by about a factor of 2 or so. So furthermore, and this is work we really just published last fall, we really definitively proved, I'm probably not going to go through the data in too much detail unless somebody has questions about it, we actually definitively proved that this molecule was functioning kind of on the granddaddy stem cell. So this molecule directly signaled to the very earliest, most immature stem cell and told it to start differentiating into a neuron. So what we're very interested in now is a couple of questions. Do these signals that we discovered and other people have discovered other signals that regulate various steps in the life and differentiation of this stem cell, do these mediate the effects of exercise or learning or environmental enrichment on this process of generating new neurons? And second, can we actually use these as drug targets? Can we actually use these as molecules to stimulate this process of generating new neurons in situations where the brain is not doing it well enough, such as in Alzheimer's disease or in Parkinson's disease, or even in an aged organism where all of a sudden the capacity to create new neurons begins to decline or to decrease. So I'm going to close on a couple of pieces of new data in which we've actually begun to use these molecules as ways to create new neurons in the brain for therapeutic application. So I showed you this slide earlier. There are many situations in which specific populations of cells in the body can die, and this leads to a variety of diseases, including diseases within the nervous system like Alzheimer's or Parkinson's or Huntington's. So the question is, can we actually harness these signals that we've discovered in order to be able to create new cells or new neurons? So let's pretend now that this is our astrocyte, this is our neural stem cell, and we want to kind of drug this interaction between the two. We want to trick the stem cell into thinking it's in contact with a neuron so we can induce this, trick the stem cell into thinking it's in contact with an astrocyte so we can stimulate the generation of a new neuron from this stem cell. So it turns out that not just one of these ligands, of these molecular signals on the surface of the astrocyte binds to an antenna molecule or receptor on the surface of the neural stem cell. You actually need a cluster. It's kind of a nanoscale aggregate of these receptor ligand pairs to form. And so as a result, this is something that's kind of difficult to drug. It's kind of hard to induce the aggregation or the assembly of these receptors. So my background, as was mentioned, is in chemical engineering. We do a lot of chemical synthesis. So we created a molecule that looks like this, where this is a linear polymer, just a long chain of molecules. And we attached onto it, kind of like beads on a string, these ligands, these Ephrin molecules, these biochemical signals that make neurons. So that we can then kind of infuse this or introduce the stem cell to this signal and trick it into thinking it's in contact with another cell that would tell the stem cell to turn into a neuron. So this is the chemistry that we utilize to do this. And furthermore, this is some slides or some images telling us that we've actually clustered the receptor on the surface of these neural stem cells. So shown here in red are actually dots or aggregates of those antenna receptors on the surface of the neural stem cells. And the more that we aggregate the receptor, the more signaling that we end up stimulating the stem cell with. And furthermore, the more that we aggregate these receptors, the better we are able to actually generate new neurons within cell culture. So this is the fraction of neural stem cells within culture that actually turn into neurons. And the better that we aggregate this receptor, the better we are able to control the differentiation of those cells into neurons. And finally, when we take this engineered molecule and infuse it into the brain, compared to kind of the old-fashioned way of stimulating this molecule, and if you analyze the number of newborn cells that are shown here in red that acquire a marker telling us that they've turned into neurons, we have many more in this situation compared to our control. So we're able to significantly increase the ability of these neural stem cells to turn into neurons by introducing this molecule into the brain. Yes?
Are you increasing the total number of differentiating cells, or are you just shifting cells that are differentiating anyway towards neurons?
That's a great question. Yeah. Give me a slide or two, and if I haven't answered your question, let me know. Okay, so what we've done here now is that we've introduced some material, some polymer, and are actually able to kind of control the behavior of the stem cells within this region of the brain called the hippocampus. And the hippocampus, it turns out, is ground zero for Alzheimer's disease, the hippocampus and the cortex. So this is a valuable, potential valuable thing to do, to create new neurons here. Unfortunately, we come back to Ramonica Hall now, and the majority of the brain does not actually harbor stem cells. So the cortex is also afflicted by Alzheimer's disease. There are no active stem cells here. The striatum is afflicted by Parkinson's disease and Huntington's disease. There are no stem cells here. So can we actually apply what we've learned about these molecules that stimulate stem cell function in order to maybe introduce some molecules over here that will either perhaps kind of convert some of the local cells into stem cells, or perhaps even actually recruit stem cells from other environments, and we'll create a microenvironment or a niche here locally that can generate new neurons in regions that don't typically do so. So what do we need within our little microenvironment? What would the ingredients be? Well, we need a signal that can turn the neural stem cells into neurons. But if you just add that signal, you actually deplete your neural stem cells. You take all of your potential to create new neurons, and you tell all those cells all at once, turn into new neurons, and then all of a sudden you've lost your source of new cells. So perhaps you actually need another signal that will promote the cell's proliferation, actually cause them to divide and differentiate at the same time. So I showed you this work earlier where we actually were able to discover a signal that stimulates the proliferation of the cells. And I'm going to skip past this. We incorporated this molecule into a polymer as well and added it together with this one. And then finally, I mentioned to you earlier that most regions of the brain don't actually harbor the stem cells, but we've learned a lot about signals that actually cause those cells to migrate over long distances. And so if you add this molecule, it acts kind of like breadcrumbs to attract the stem cells out of their natural microenvironments and get them to come to the point where we're injecting these signals or these materials. So if we add these three signals together, and shown here is the number of newly divided cells within a region of the brain that does not ordinarily generate new neurons, the striatum, the region of the brain that's afflicted by Parkinson's disease and Huntington's disease, we stimulate the number of newborn cells, and importantly, if you wait a certain amount of time, we actually are increasing the number of newborn neurons by about a factor of 8 within those regions of the brain, or within that region of the brain. So I think I've shown you an example where you can start all the way from a basic observation that these cells exist within the brain, learn something about the molecules that control them, and then apply that knowledge to actually generate new cells or new neurons in regions that don't ordinarily do so. So we're hoping to take steps towards clinical application and cell replacement therapy. Yes?
Since this neural creation in the brain is rare, what's happening to the regions of the brain that this substance is drawing upon?
The regions of the brain that this substance is drawing upon? Mm-hmm. So those cells, they do have the ability to self-renew. So meaning if you actually, going back earlier, I showed you that study where investigators figured out a way to actually ablate or kill a lot of the neural stem cells within the brain, and that had the effects on learning and memory. That was how it was proven, really, that these cells play an important role in learning and memory. If you then allow the animal to recover, even though its stem cells have been largely depleted, it'll repopulate. So that's one of the beauties of stem cells. They have the capacity to self-renew or to maintain themselves for long periods of time. So if we steal a few of them from the hippocampus or from the subventricular zone, those regions have the ability to self-renew or to repopulate the stem cells. Yes?
So today, what are the chances, according to what you mentioned, like to manipulate the memory of the brain? What are the chances?
So we haven't taken a look at, so there has been one study, so I'm probably not going to go back that many slides. So I showed you that kind of progression where you start out with a neural stem cell and then differentiate into a neuroprogenitor and then differentiate into a neuron. It turns out that, and I showed you earlier, a lot of the cells that kind of start down that road actually die along the way. So they don't permanently contribute to your brain. So one of the molecules that makes them die is a molecule called a caspase. It's a molecule that's involved in kind of cells committing suicide in a very programmed and intentional way, a process called apoptosis. So a couple of years ago, investigators actually overproduced an inhibitor of this molecule within the neural stem cells. And as a result, they kind of prevented the neural stem cells from dying. And that greatly increased the number of newborn permanent neurons that got incorporated. And those animals were smarter. So they performed better on tasks associated with learning and memory.
And can you inject like these molecules during before the birth process, like during the creation process, or after birth? Like which would probably the better process to do?
That's an open question. It sounds like a good study to me. But I would say now, that had been done within a mouse. You can create a smarter mouse. We're probably not interested in manipulating learning and memory, but we're very interested in treating human disease. once you start talking about making smarter people, then that begins to cross into sort of bioethical questions. Yes?
In your earlier slide, you showed that you enriched the environment of the mice by putting these pipes in. And I'm wondering, we're in a learning environment here, and it's an enriched environment. Do you have any insights into, like, say, does reading a book enrich the environment as much as a video game or anything like that?
I don't know if that's actually been addressed or if that's been studied. So that maze study I showed you earlier, where the animals learn their way through the maze, and as a result, the amount of time it takes them to get through it decreases over time. So it's been shown, at least in some studies, there's some conflicts within the literature, but it's been shown that the very act of learning actually stimulates neurogenesis. So people who are actively engaged with their environment and will probably, if we believe what the mouse tells us, will actually implicate or change the process of neurogenesis within our brains. So for what it's worth, it's been shown, for example, in Alzheimer's patients, if patients are actually encouraged to learn, to read, to do crossword puzzles, to go through kind of learning and memory tests, brain teasers, it'll slow the progression of the Alzheimer's disease. So it's an open question as to whether or not this neurogenesis I told you about plays a role in that process, or whether that's simply acting on kind of the mature neurons. But it's suggestive that potentially neurogenesis could play a role. Hey, other questions? Okay. Well, with that, I just wanted to leave you with a couple of bullet point concepts, and then I'd be happy to open up for more discussion. So neural stem cells exist in each of our brains. And it's the idea that the adult brain is something fixed, ended, and immutable is actually myth now. The adult brain does have the ability to create new neurons throughout life. So it's very actively, these cells are populating the brain during development, and they persist throughout adulthood. So this process of adult neurogenesis is also very dynamic. It's controlled by our choices, by our behaviors, by our diet, it turns out as well. And this provides the brain with a very dynamic means to regulate the function of adult learning and memory. And furthermore, this process of adult neurogenesis is regulated by molecules at the heart of it. And the more we learn about those molecules, the better opportunity we have to actually turn them into therapeutics, to enable these cells or endow them with the ability to do things that maybe nature didn't intend them to do, such as repopulate the brain from the effects of an injury or from Parkinson's disease. So I'd like to very quickly acknowledge the people who did the work that I showed you that came from our own lab. So the work on sonic hedgehog was conducted by Karen Lai. The work on this Ephrin molecule was discovered by Randy Ashton and Anthony Conway. And finally, the work on introducing these signaling molecules into the brain, both in the hippocampus as well as other regions, to stimulate the process of new neuronal generation was conducted by Anthony Conway. So thanks a lot for your time, and I'd be happy to answer any questions.
I was just wondering if you're aware of any studies that have to do with the effect of substance abuse on genesis?
Yes, it's been shown that within rodents at least, that excessive alcohol and things like cocaine reduce neurogenesis. So we have all these correlations, right, between behavioral choices and things that really adversely affect brain function. So it has not been definitively proven that some of the bad effects of cocaine on the brain are mediated through this reduction in neurogenesis. But again, it's a very suggestive correlation.
Feedback loops are really important in all kinds of information theory. It seems like there's no closer thing to the information theory for us than the human brain. Within your own group, do you find that you can increase the feedback loop of the iterations of improvements of knowledge and how you cycle it back into your own learning process? It looks like you've done a lot of creative work. Do you have a strategy or something that enables you to do top-flight research?
Well, I mean, we certainly every year try to get better and better at what we do. I'm not sure if there's a formal process by which we do that. I will mention kind of one interesting anecdote, which is that there's, if you actually consider just memory, your ability to memorize something, which is a certain kind of memory, You know, if I spat out 100 digits or numbers at you, how many could you remember them? If I showed you 100 faces and names, how many of those could you remember later on? So there's a kind of a rather famous mnemonic within the memory field where the hippocampus actually encodes spatial memory. So that maze is something that proves to us that the hippocampus and these neural stem cells are important for our sense of spatial memory. So we, as human beings, probably due to evolution, you know, our need to find our way back to a food source or find our way back to the cave, have a very well-developed sense of spatial memory. And there are other types of memory tasks, like, you know, memorizing a list of digits that we need now, you know, perhaps as a computer scientist or somebody who works with Microsoft Excel, that evolution didn't really teach us how to do. You know, our brains are not very good at those types of things. So one mnemonic that people utilize is to actually use the spatial memory, which is really good, in order to encode other types of memories. So for example, imagine you're walking on your way home and you've done that 100 times. You know that very well spatially. Let's say you deposit a digit. You know, once you pass this mailbox, you put the number 2 on it. Once you pass your friend's house, you put the number 10 on it. You can actually embed other types of memory inside of spatial memory and then walk through the neighborhood to be able to recover the digits. So that's a very common mnemonic technique that's utilized within the memory field, to use one type of memory that we're very good at to encode or to remember things that we're not so good at remembering.
I'm going to take a second to get back to you.
Yeah, so I understand that you're in bioengineering, and you know you're sort of more on the synthetic substance side of things. But I wonder if you have anything to say about alternative methods of doing the same thing, like for example, meditation. I know that there's some research that confirms that there is the potential, if not the actual producing of new neural cells as a result of meditation.
I'd love to hear that. I'm not familiar with that work. A lot of what we know comes from studying mice and rats, from studying animal models. And it's, so whatever kind of experimental input, you know, behavior, enriched environment, or sleep cycles, or diet, or things like that, that you can kind of experimentally test with an animal, then you can develop, you know, kind of knowledge about how that affects newborn neurons. It's, you know, really hard to do that with adults. Because
so you're only researching things that can essentially be seen.
Yes, it's hard to do that with human beings, obviously, because
right.
We need to be able to measure things in systems where we have control. And it's hard to probably teach a mouse how to meditate.